Hello, everyone. Welcome to today's event. I'm Brian Domansky, Strategic Collaboration Scientist at Shimadzu Scientific Instruments. I'll serve as your host for a discussion about increasing productivity for HPLC tasks in pharma and biopharma QA/QC. Before we start, I want to share a couple of notes for our viewers. The webinar console has a variety of items to help enhance your experience and interaction with us. In the screen, you'll see the following items. The slides will appear in the center of the console. Directly under the slides, you will see the related content list with clickable links relevant to the material being presented today. On the top left of the console is the question widget. Please submit your questions during the presentation through this widget, and we will answer them during the Q&A session. Just below the question box are survey questions that you may fill out anytime during or after the presentation. If you would like to be contacted by a Shimadzu representative directly, use the Request Demo section on the right to start that process. The icons at the bottom of the screen can be used to restore any of the widgets in case they are minimized or hidden. All right, let's get started with today's presentation. Our speaker will be Kevin Wands, who currently serves as the Marketing Manager for Pharma and Biopharma at Shimadzu Scientific Instruments, and has more than 9 years of experience in biologics characterization and neuroscience research. We welcome Kevin to present how built-in analytical intelligence functions in HPLC systems accelerate productivity in QA/QC labs.
Thanks for the intro, Brian. So let's get started. So Shimadzu has two different types of LCs. On the left is our integrated type, the I series, and on our right is our modular new Nexera series. Today I'd like to spend time highlighting our I series line and how it can be the workhorse for any pharma lab, whether you're in QA/QC, if you're developing an LC method, or if you're in any GMP setting or regulated environment. The I series has had a few different iterations, but now it comes to you with some of the most advanced features on the market. We have flow pilot function, we have the iPeak finder to help with data processing. We can all agree that data processing is one of the most time-consuming processes to any pharma workflow. So I'd like to show you with today's webinar how iPeak finder and iPDEA can help ease your workload. And of course, we also have our Lab Solutions MD, which is our method development software for analytical R&D labs that come with AI functions to help you with the method development workflow. Now, Shimadzu will say our HPLC systems are as smart as a human being, and that's due to the combination of AI and automation that we use to improve efficiency of pharma workflows that we like to call analytical intelligence. Analytical intelligence is our key differentiator to maximize lab throughput, generate high data quality, and to increase overall lab efficiency. How does it maximize throughput? I'll give you some examples. First is our mobile phase monitoring function, which is like having a balance on top of your LC system to measure the real-time volume of your mobile phase. We've all been in that situation where we've had these long sequences of samples to run on an LC, and you aren't sure if you made enough mobile phase to run all of your samples. Well, our MPM device is there to help tell you how much volume you need in the first place. We also have the flow pilot function, which helps you increase your flow rate gradually. So if you're running at a high flow rate, or if you have a small particle size column, your column stays protected by our flow pilot function to ramp up flow rate slowly. And it works in conjunction with our column oven, so the temperature stays in check as well. Not to mention, you can access your system even when you're not in the lab. And of course, we have our iPDEA function, which is our intelligence peak deconvolution analysis. This is the feature that helps you separate those tough to separate peaks, which we'll discuss more later. And overall, our I series can take care of itself by telling the lab manager when the system is due for maintenance, and it will also preempt the manager whenever the system needs to change any consumable parts. It has the automation functions of intelligent startup and shutdown functions at preset times. So when you want the I series to wake up and when you want them to shut down after you register your batch sequence, it'll shut down automatically. While these features are beneficial for not only QA/QC, the features of the I series can also benefit R&D labs as well. Say you're in an R&D lab and you're developing an LC method that will eventually be tech transferred over to a QA/QC lab. The I-Series, it's meant for you. But for anyone that's watching in a QA/QC lab, I'm sure you're familiar with USP 621 changes that happened back in December of 2022. USP, they used to be pretty strict on gradient changes, but after some amendments made back in with the amendment in December of 2022, they've loosened regulation to allow QA/QC users to change their gradient methods. USP 621, it basically defines the acceptable method changes without fundamentally modifying the USP method. As long as the user fulfills the criteria as shown in this table on this slide, changing to smaller column dimensions to shorten the analysis time doesn't require the revalidation. However, converting from HPLC to UHPLC with the myriad of parameters that you have to modify while staying compliant could be tricky. Luckily, Shimadzu has a solution that we'll discuss later. Here we have an example of a common USP monograph of budesonide. Say you have this 20-minute method, and you want to run the method in half that time. These are the calculations that need to be done to ensure that you follow USP Chapter 621. But what if I told you there's a program that calculates all of this for you? I know not everyone watching this webinar is a math whiz, so I can assume a lot of you would be more than relieved to know that you don't have to do these calculations. Here we have the method transfer tool, which adds in the L over DP calculation. So if you have your existing HPLC method and you want to optimize it to make it a faster method, you can just set it up on the method transfer tool to set what the UHPLC column that you want to use. Then the software will automatically tell you whether you follow and ensure that you are within criteria from USP 621. Also, say you're converting a gradient method, which you'd have to calculate all by yourself. Sounds like a tedious process. And that's why Shimadzu's method transfer tool can transfer gradient methods or from any third-party vendor as well. As I mentioned earlier, the system has an auto startup function. Say you start your work at 8 in the morning. Great. You can set up your I-Series to start up at 7:45 a.m. and get to immediately equilibrating. Therefore, once you enter the lab, you can put in your standards and your samples, and you can start your analysis immediately without having to wait for conditioning. Analytical Intelligence also features automated startup where the LC will not only set up startup, it'll also start automatically purge, equilibrate, and heat up the column. Also, we have our mobile phase monitor function for mobile phase monitoring, which is our patented function where you now have a balance on top of your I-Series. So if you register your long batch of samples and you prepare mobile phase that's not sufficient to finish your batch, there will be a dialog box that pops up and lets you know that the mobile phase you prepared is not suitable, and it'll tell you to either prepare more or reduce the number of injections from your sequence. This can help you prevent system dry out. This helps prevent this prevents column damage, and it prevents air bubbles from entering the system. Also, everybody has a different analysis method that requires a different mobile phase setup, which means different amounts of different solutions going through your I-Series. But luckily, you can mix and match your bottle setup right on your I-Series, just as long as it fits in the right holder. This is especially helpful if you have a gradient method monitoring multiple mobile phase bottles at once. The next feature I'd like to elaborate more on is the flow pilot function. This is another patented technology that works in conjunction with the column oven, so the chemist does not have to sit at the instrument and increase the flow rate gradually. This protects your columns, which we know are not cheap. So FlowPilot will gradually increase the flow along with the temperature to prevent pressure jumps, which will extend the life of your HPLC. Not to mention, we have a state-of-the-art auto sampler that can hold up to 216 vials, as well as different types of plates in the same auto sampler that has automatic recognition. So it'll recognize the plate rack you've entered into the auto sampler, and it'll configure the plates based off of the rack that you enter. So So obviously a QA/QC lab requires the utmost reliable results. So it's very important with the iSeries auto sampler to be very robust and to ensure ultra-low carryover. For example, if you inject a high concentration sample, such as this 5 gram per liter injection, followed by a blank, the instrument is capable of 0.0025% carryover. iSeries also comes with an extended column oven, which allows you to store up to 6 short columns and 3 long columns. So our ovens have the capacity to even install a switching valve so that you can switch among methods automatically in the oven. We also have extended the oven temperature to room temp minus 15 degrees. So let's say you're analyzing a USB monograph that has to be at 10 degrees Celsius. Our low temp option can achieve that for you. Not to mention, the iSeries comes with a front panel that allows you to set the parameters directly from said panel. And in case you want to do some troubleshooting on the system itself, even the end user can also use any smart device and scan the QR code on the screen and find out how to do the end user maintenance. For example, how to clean the suction filter of the mobile phase. This front panel allows you at a glance to know what's going through your analysis, whether the system is running or if it's in standby mode, so you can easily see through this slide. We also have many lab solution software functions that can really make your lab work for you. First, being automated sample pretreatment, so you can auto-dilute to replace manual dilution with our auto sampler. Next, the iPeak Finder, which will help integrate challenging peaks, making data processing a breeze. Then we have seamless method transfer integrated into Lab Solutions CS. So if you want to convert that Shimadzu LC method into a HPLC method, this program can be used for that. And of course, we have IPDEA, which can resolve the unresolved peaks like what is shown in the screen now. Now, sometimes let's say a drug has to be pre-treated with a solution before it's ran on an HPLC. But that treatment can lead to its oxidation or overall degradation in the process. Our iSeries is equipped with an automatic pretreatment function as a standard, which can be used for sample dilution or reagent addition. Therefore, you can carry out procedures such as derivatization right within the needle of the autosampler, avoiding a derivatized sample sitting on an autosampler potentially degrading before it's injected on the machine. Here I have some example images, one being the pretreatment settings screen seen on Lab Solutions, displaying the reaction setup in the needle, and an example chromatogram of methionine and cysteine standards that were oxidized to methionine sulfone and cysteic acid, respectively, on the iSeries and analyzed within 12 minutes. So in the auto-sample pretreatment function, you can program the needle to add a few reagents to derivatize your amino acids, and then it'll mix within the needle, and then a derivatized sample will load onto your system. Manual derivatization can lead to degrading before it gets ran on the HPLC, and this pretreatment function can eliminate that risk. When it comes to peak integration across a long set of samples, it's all about consistent integration across those samples. And iPeak Finder can help maintain that consistency. By adjusting peak detection thresholds, integration range, and baseline processing, reliable peak data can be acquired regardless of the user's operation skill. Obviously, sustainability is an important consideration in optimizing pharma workflows. And one way to do that is to convert a conventional HPLC method into a UHPLC method to give it a shorter runtime, so you can use less organic solvent. But there are so many parameters that you need to consider, such as flow rate, sampling rate, delay volume, injection volume, gradient program, so many moving parts, if you ask me. Now, we have an integrated method transfer function that can help you convert the method seamlessly in the software with Lab Solutions. You just click the method transfer button, open up your existing HPLC method, then key in the new column dimensions that you would like to use and the UHPLC column dimension you need, and then click Convert. The method will then convert seamlessly, including the gradient program, and then you can save it as a new method. one key takeaway that we want you to take away from this webinar is that methods can be converted seamlessly in lab solutions for UHPLC analysis or for HPLC. For example, let's take this method transfer for an antibiotic profile that runs for 40 minutes on HPLC. And you want to shorten that to a UHPLC method so it reduces your analysis time to a much shorter time. With the method transfer tool on Lab Solutions, that 40-minute method has now been shortened to a 12-minute method, all from a few clicks. Now, onto IPDEA. Sometimes we have to analyze the mean API, but we also need to check whether it coelutes with any impurity. So, if you suspect that is the case, you can use IPDEA to check. This is to confirm that in fact there is a coelution, and not only does the software help separate the peak, but it also helps separate the UV spectrum. So this is available when you use our PDA detector with Lab Solutions. Now some of you may wonder how many peaks can be separated with this PDA function. And the answer is up to 5 peaks. They're able to be separated using this function. Now I also want to talk about our method scouting systems that are for integrated and modular systems. They can eliminate the manual column and mobile phase changeovers. One of our most popular systems is the modular Nexera series method scouting system. It has the capability to test with 8 types of mobile phases and up to 12 different columns simultaneously. Also available in the integrated I-series systems with lesser number of solvent and column capabilities. Our method scouting systems have automated solvent blending capabilities to eliminate time-consuming, complex mobile phase preparation by the users. So I mentioned it before, but now I'll elaborate more on it. I'd also like to mention Lab Solutions MD for our R&D labs. It's packed with automation and AI to shorten method development time, and it also comes with automated peak tracking to save time and labor during data processing, and it can achieve high efficiency, so it reduces time spent on method development. Now, the I-series may look compact in size as we are integrating so many aspects of a standard HPLC into just one box, but it actually lends itself to being a very flexible instrument, what with its capability of working with an array of detectors in addition to the UV PDA detector already built into the system. It can work with a refractive index detector, a fluorescence detector, it can work with evaporative light scattering detector, and several mass spectrometers. Now, not only is the I-series compatible with multiple detectors, but Shimadzu also has compatibility with a lot of third-party vendors and LC systems as well. With the Smart Kit, you can do easy method transfer from any LC model, even from other brands. So data can be easily converted from other third-party vendor systems or Shimadzu's own legacy systems right onto our very own I-series. Ultimately, when it comes to analytical intelligence, we at Shimadzu want to bring you flexibility and the ability to operate your instrument from anywhere, whether it be in the lab or in the office. Finally, we want to leave this webinar stating that our I-series is the industry standard that is as intelligent as a human working in your own lab. And we've stacked the I-series with tons of analytical intelligence functions to automate your workflow and make your day in the lab much, much easier.
Let's thank Kevin for such an engaging presentation. As we prepare for the Q&A session, I encourage you to submit questions for the discussion through the question widget in the upper left of the console. To learn more about how I-series HPLC and UHPLC solutions can increase efficiency with analytical intelligence, check out the related content list. If you have not had a chance, please fill out the survey questions on the left-hand side to provide feedback for us and request additional information for you, including the ability for you to sign up for the Shimadzu e-newsletter. This once-monthly blast includes information regarding upcoming webinars and events, encompassing a wide variety of application areas.
At this time, we will begin our Q&A session. Thank you to the audience for attending, and I welcome you to still submit any questions that you may have. We do have time to cover a few. One of the first ones that has come in is in regard to method transfer. And the question is, how does method transfer achieve higher reproducibility?
Sure thing. So the method transfer tool incorporates an analytical condition transfer and optimization, or ACTO. It's a function in lab solutions where users can transfer their injection timings matched to differences in the system volumes between instruments, and so without editing the concentration gradient programs in existing methods. So reproducing analysis differences between systems can be difficult even if you use the same column and mobile phase conditions, but gradient analysis times can differ due to differences in internal volumes between LC systems. However, with the ACTO, the differences in internal volume can be fine-tuned to heighten data reproducibility between different systems, streamlining your method transfer.
Excellent. There was another question that had to do with analytical intelligence functions and the peak finder algorithm.
Oh, sure. So when you optimize your instrument and your column to enable faster analysis speeds and higher throughput, there's still a lot of data that requires processing, and manual peak integration is often the bottleneck. But with iPeakFinder, it makes it possible to perform a large amount of integrating peaks, including shoulder detection, even in complex chromatograms that previously would require manual peak integration. It only requires 3 simple parameter settings in your conventional method: baseline processing, peak integration range, and peak detection threshold.
Excellent. There's another question that just came in regarding the slides and them being shared, and I'll actually speak to that. We would ask that anyone who is interested in having a copy of these slides, please reach out to either Kevin Wands or myself, and we'll be happy to help you obtain a copy of those. You can also reach out through your local Shimadzu representative. We have the information if you submit a question here that we can send it to you directly. So we would encourage you to reach out, but we will send you a copy if you've asked for one in the question section during the presentation. There's another question, Kevin, related to the IPDEA function, that the photodiode array deconvolution function. Can you tell me a little bit more about that?
Sure thing. So it uses wavelengths and time ranges. That's how it can deconvolute multi-component peaks that provide both absorbance versus time and spectrum data for each feature identified by the raw data.
Excellent. All right. We have another question that has come in, again, another request for the slides, and I welcome anyone who's viewing the presentation right now. If you would like a copy of that sent directly to the email you used for registration, go ahead and throw a comment in that question widget, and we'll make sure that you get a copy of those. And I again want to thank Kevin, our speaker, for his excellent job today. We've reached the end of our time for our allocated webinar. We thank you for all these interesting questions. We don't have any additional time to answer those here online, but we will reach out to anyone who puts a comment in the question section now or at any time in future. If you're watching this on demand, we'll send you a direct answer to your questions. Thank you for attending and participating, and an email link will be sent to your email used for registration with the uploaded version of the recorded presentation that you may view again at your leisure. And we encourage you and your colleagues to attend our upcoming webinars and use the survey to engage with the Shimadzu e-newsletter. Thank you and have a wonderful day.